Perfect. Well, hello everyone. Thank you for coming to my talk. My name is Logan. I'm a fourth year, or going into my fifth year PhD candidate in the Bernal Lab at Auburn University. I'm excited to tell you about my research on thermal tolerance of cryptobenthic fishes. So as I'm sure we're all aware, climate change is happening. It's a very hot topic, but it's responsible because of an increase of greenhouse gases in our atmosphere. We all know Earth is warming, but what's important is we have a lot of future scenarios of this warming. So if we have no cuts in emissions, we're going to see this very rapid increase in warming temperatures. And there's some lesser scenarios where we do make some cuts, but even in the best case scenario, even if we do drastic cuts, just based off of the concentrations we have, we're still going to see an increase in warming. So understanding how organisms are going to be impacted by minor thermal stress under future conditions is a very relevant question to ask. Now, mean temperatures are going to increase, but it's not going to be even across space in the globe. As you can see in this graph on the right, under one of the worst case scenarios in the Gulf of Mexico and the Western Atlantic, different regions of water are going to increase in temperature at different rates. So kind of understanding what organisms in space are going to experience is another important question. Now, thermal stress on fish has been kind of well understood, but mostly in species of commercial importance, so things like tuna, snapper, big fish that we care about because we eat them, they're good for trade and nutrition. And a lot of this has been done on things like immunology, metabolism, transgenerational effects, but less work has been done on lesser known species such as cryptobenthic reef fishes. What is a cryptobenthic reef fish, you might ask? Well, they're very pretty, and there's a lot of them. But if you were to ask Brandel, who's kind of the godfather of these fishes, it has to be a fish. It has to be associated with a benthos, so some sort of structure. So normally these are found on the bottom, but you can find these on boat pilings, docks, anything where there's structure. So between barnacles, oysters, corals, sponges, they're all over the place. And they have to be behaviorally or morphologically cryptic, so they blend in with their environment either by how they look or how they act. And they have to be small, so normally less than 5 centimeters total length. And I want to make a point here that some people, or I normally forget to make, is that this isn't just one evolutionary branch of fish. This is many different families that have cryptobenthic species within them that kind of occupy this cryptobenthic micro-niche. So they're pretty, but why should we care about them? One of the main things is they're very important for biodiversity. On coral reefs, they make up about half of the fish species biodiversity there, and they make up or account for about 60% of the consumed fish biomass. So they're very, very important for nutrient cycling and making up that kind of lower level of the food web to support those higher level fishes that we care about. Just on this graph here, we have cryptobenthic reef fish families in blue and larger fish families in white. And those three families, the gobies, the blennies, and the cardinal fishes, make up about 60 to 70% of the larval pool for fishes. So a lot of that consumed fish biomass comes from this massive push towards the larval pool. So a lot of my research is focused around what's a fish to do under climate change and how they're coping with it. So some of the aims of this project I'm going to talk to you about are measuring CT max, which is identified as a loss of equilibrium. We'll get to that in a second. CT leth using intermittent automated respirometry, kind of understanding their metabolic performance. And then we're going to compare these across sites in the northern Gulf of Mexico. So these are our 3 sample locations. We have Port Aransas, Texas, Dauphin Island, Alabama, and Marineland, Florida. You'll notice that 2 of them are at the same latitude, and Port Aransas is about 2 degrees south. We have a slight latitudinal gradient. But we should have some, or at least one known geographic barrier to dispersal or gene flow, and that's at the southern tip of Florida where there's a lot of freshwater runoff from the Everglades. So we have populations that could be genetically disconnected, so that could be leading to some sort of difference. So for CT max, it stands for critical thermal maximum. This is our fun little setup here, but we measure this by incrementally increasing the water temperature by about 1 degree Celsius every 10 minutes. And CT max is measured as a loss of equilibrium. So if you think of a fish swimming around, when it gets too hot, it'll start to peter over and is no longer able to right itself. So that's what defined as our endpoint. I want to heighten that this is not a death temperature. We're not just boiling the fish until they die. But we kind of consider this an ecological death. They lose their locomotory ability. So if there's a bigger fish that comes by, they can't swim away from the predator. If the water's getting too warm, they can't swim away to somewhere cooler. So it's kind of that ecological death temperature. If you do lower the temperature after they hit this point, 95% of the time they can recover, and you could repeat this. But we just did one measurement for individuals. They're cryptobenthic species. They're very cryptic, so a lot of the species look very similar. So we used, we pulled out mitochondrial genes, specifically CO1, to kind of confirm these IDs. So from our results, we heated up a lot of fish across these 3 sites, almost 300. We have some good comparisons between all 3 sites, but I'm going to talk about just the community we found in Alabama. So we're going to go on a trip to sunny, beautiful Dauphin Island, and this is what our data kind of looks like. So these are density plots kind of overlaid on top of each other. On the x-axis, we have the CT max temperature in degrees Celsius. This is where they were hitting. And then for each individual y-bar, that is a species. And the higher the peak is and the thinner that peak is, that's more individuals hitting CT max at that temperature. If there's a wider part towards the bottom, that's just more variation in the data. So with this graph right here, just in our Alabama population, the top 4 species are all blennies, and the bottom species are gobies. So clearly the goby has like the highest CT max within this community. And some of these fish, especially Hypsiblennius envimar here, has a very low CT max, about 33 degrees Celsius. So under future conditions, Dauphin Island gets to about 29 to 30 degrees Celsius on the hottest part of the month, or the hottest part of the year in August. So under future scenarios, we could be seeing potential shifts in this community just from these measurements right here with cryptobenthics. So kind of interesting, but also kind of scary. So that's just for Alabama. If we look at 3 species that we could see from all over, we'll look at the gobies first. We have Texas having the highest CT max, just over 40 degrees, which is significantly different than Alabama and Florida, which aren't different from each other, but it's slightly lower. For one of the blenny species, Scartella cristata, we see a different trend. Texas still has the highest CT max, but we see differences between each location. So within these 2 species, we didn't really see a lot of structure between sites. Again, this is take this with a grain of salt. This is just mitochondrial CO1. So we're looking at potentially using whole genomes to kind of tease apart what this difference might be responsible with. Next up is Hypleurochilus. There's 2 species here. This has kind of been a problem that we've been trying to address, but we recently have kind of teased apart. So these 2 species, Hypleurochilus multifilus and Hypleurochilus geminatus, they were described in the 90s, and it was said that they had overlap across the ranges that we measured. And as we found out, that's not true. So Hypleurochilus multifilus is only found in the Gulf of Mexico, and Hypleurochilus geminatus is in the Atlantic coast of Florida. So we do find differences between our Alabama and Texas sites for Hypleurochilus multifilus, and geminatus is kind of on its own. But when we add them all together, we see that there is a similar trend to the other blenny Scartella cristata, with Texas having the highest CT max, followed by Florida and then Alabama. So I think I skipped past, yeah. So we do see some structure between our Atlantic and Gulf populations, but between our Gulf populations, we don't see that. So that kind of supports that they are definitely different. So those are our CT max measurements just from seeing that loss of equilibrium. But another dimension we wanted to see with thermal tolerance is kind of how it's affecting their metabolism. So we're measuring that with respirometry. We have 2 kind of theoretical frameworks of ectotherms, like their metabolic performance as temperature increases. So in the first one, we have a pretty sharp peak followed by a gradual decline as a fish heats up too much. And in the second scenario, we have this kind of gradual increase before a very, very sharp crash, and we believe that that's where the CT max number is. So we're adding this to kind of bolster our measurements for thermal tolerance. So again, we went back to Dauphin Island, and we have measurements from that community. This is our setup here, which we had to tweak a little bit, which was a lot of fun. But each of those chambers along the bottom can have a fish in them, and they're set up to 2 pump systems. So one pump system is open circuit, and we can flush the chamber with fresh oxygenated water. The second one is closed circuit, and we can turn off that open circuit pump system and then measure the oxygen in the closed system. And as the fish is respiring, we can watch that oxygen concentration decrease over time give it more fresh water so it can recover, and then do multiple measurements. So in this case, we did a slower incremental temperature increase, about 2 degrees Celsius an hour. That allowed us to do multiple replicate measurements at each temperature point in time. So this is what our data looks like plotted. So on the y-axis, we have milligrams of oxygen consumed per gram of fish per hour, and on the x-axis, we have temperature. So we have our goby species in red and our 2 blenny species in blue and green. I've tried to play around with ggplot a lot. For whatever reason, the goby line isn't going down, but I want to confirm that they did crash. We did get these to their CT max. So if you are a whiz with ggplot, please talk to me. But this is kind of an interesting thing where we see that trend where we have a gradual increase in metabolism as temperature increases, but then we do have a kind of crash that happens afterwards. And if we plot the CT max averages from our Alabama population, you see that they're all kind of towards that trend where it's beginning to crash. So the CT max and the quote-unquote faster trend is hitting around that point in time. So again, the CT max measurement isn't when they're dying, but we're getting kind of at that peak where they can no longer function efficiently. So why did we boil all of these fish? Well, current high temperatures, like I said, are 3 to 4 degrees Celsius lower than some of the species CT maxes that we measured. So they're pretty consistent across sites, but from what our measurements are, they're kind of within the danger zone of future climate scenarios, just with that average heating range. We think that gobies are definitely going to fare better than the blennies in the Gulf, just from our measurements. There's tons of other species out there, but just using ours as a metric. And then we're really interested in see what is driving these differences in thermal tolerance between these locations. So could it be the developmental temperature they're exposed to, like when they're born, when they spawn, and then again, if it's genetically controlled. So you may have seen some other headlines like this with these crazy heat waves that have been happening in the Gulf. So just last year, it got to above 36 degrees Celsius, which was higher than almost every blenny species we measured. So that's kind of spooky. So again, the average temperature increase is bad, but we're going to have these extreme events too, which could be really chaotic and bad for some of these communities. So one thing, kind of a plug of an idea that I have, kind of like marine protected areas where we're taking these parts of ecosystems that are very, very important for conservation efforts. I think including models and data like this into a model of fish and other kind of marine species across space where we know there's going to be differences in that temperature increase could tell us good things about conservation efforts for the future. So if you have any ideas about that, I'd love to talk with you. Thank you to everyone who made this possible. We caught a lot of fish and had to keep them alive, so thank you all. Thank you to the fish who made this possible. Couldn't have done it without you. And thank you for coming to my talk. We'll take any questions. Yes.
Yeah, thanks. Really nice talk.
Thank you.
But also a bit freaky or spooky, as you were saying.
Mhm.
But I was wondering, so how realistic are your, or how ecologically relevant are your measures, do you think?
Mhm.
Because you obviously are quite, like, increasing the temperature quite rapidly.
Right.
Do you, like, that must play an effect? Like, do you think that they could actually survive these longer, these higher temperatures if they maybe have time to sort of forfeit it to the plasticity, or would there be some pockets of water that are?
Yeah, so temperature of the water is probably not going to increase as rapidly as we're measuring that. Within the CT max community, there's a lot of arguments of how fast or how slow you should increase the water, and that's kind of for like acclimation to that temperature, or if you're just stressing the fish out so much. So in the wild, I don't think that's relevant, and then in the wild, they could potentially swim deeper into the water or swim away. These fish, like I mentioned, they're benthic, so they can't really swim very far, but they could swim deeper. But then there's some other things like competition that they might run into in those scenarios. So I think in like heat wave scenarios, they'd probably try and move, but then the thermal stress also changes their behavior as well. So there's probably a lot of things going on there. I hope that answered your question. Okay. Anyone else?
So thank you so much to everyone for coming. My name is Anusha Bishop, and I'm a graduate student at UC Berkeley. And today I'm going to be talking about some of my work looking at the landscape drivers of genomic variation in fence lizards across California. So we know that genetic diversity is really important. The loss of genetic diversity can lead to inbreeding, which has long-term consequences for population viability. And we also know it's important for evolutionary potential, or the ability to respond to long-term change. We also know that genetic diversity is under threat, with some estimating that as much as 10% of genetic diversity may already be lost in many non-threatened and threatened species across the world. And protecting genetic diversity is inherently a spatial problem. So often in management, we may want to know, where are these hot spots of genetic diversity? Where are these cold spots of genetic diversity? And then fundamentally, what are the drivers of where these hot and cold spots are? So what actually drives genetic diversity across space? Well, fundamentally, landscapes drive variation in population size, in gene flow, and selection through time. And all of these processes through time cumulatively shape the patterns of genetic diversity that we see today. So for example, you might imagine that areas of high stability, so persistent habitat, persistent populations, gene flow, are going to have more genetic diversity, while areas that have experienced population bottlenecks, range expansions, extirpations are going to have lower genetic diversity. And we think of these processes as happening over really long periods of time, right? Genetic diversity respects the history of a lineage. So often we're thinking glacial cycles over hundreds and thousands of years. However, recent research has also shown that contemporary landscapes can affect genetic diversity. So things like temperature gradients, latitude, human impacts. And finally, other evolutionary processes, such as admixture between diverged lineages, can affect genetic diversity patterns as well. But what we know little about is how these variables all work together and contribute to the patterns of genetic diversity that we see today. So to dive into this question, we're going to California in the United States. And this is a great place to study landscapes because if you haven't been there, there's just so much variation in vegetation, in temperature, precipitation, anthropogenic climate change, human modification, and going back in time, paleoclimate stability, glaciation across the state, a bunch of variation. And then the heroes of our stories are these cute little lizards, Scoloporus occidentalis, also known as the western fence lizard. And they're a generalist lizard found almost everywhere in California. So a great species to study landscape processes because we find them everywhere across the landscape. And some great work has already been done in this species by Bouzy et al. In 2022, characterizing the demography and history of this species, where they found that they think the initial divergence in the species occurred about 1 million years ago in Southern California, and then over the next hundreds of thousands of years, they expanded northward all the way up into Washington. And this expansion coincided with a lot of glacial cycles, a lot of climate change, which makes it really interesting to study the processes, both historical and contemporary, that affect genetic diversity. So with this in mind, we used whole genome data from 162 individual Scoloporus across California, and this is part of the California Conservation Genomics Project. I'm sure you'll see a lot of other talks at this meeting with that kind of data. And we calculated genetic diversity based on whole genome heterozygosity across over 14 million SNPs. So here's a map where each individual is colored by heterozygosity, higher values, more genetic diversity. And we modeled heterozygosity as a function of both historical and contemporary landscapes. So playing for team historical landscapes, we have paleoclimate temperature change, which is temperature change over about 100,000 years of quaternary glacial cycles. We also have bioclimatic stability, which is both temperature and precipitation variation over much longer periods of time, about 1 million years back. And then finally, we have last glacial maximum glacial extent. So where were glaciers in California about 20,000 years ago? And then playing for team contemporary landscapes, we have recent temperature change, so anthropogenic climate change over the last 100 years. Then we also have the current temperature and vegetation landscape across California. And finally, human modification as estimated with the Global Human Modification Index. And one other variable we included in our model, which I want to kind of expand on a bit more, is admixture. And the way we estimated admixture is basically, for those of you who run structure analyses, this may seem familiar. We imagine the green is our landscape. We have each point is an individual, and they're colored by their ancestry. So let's imagine we have a pink population and a yellow population, and we have some individuals that are all pink or all yellow, so no mixed ancestry, and then some that are a mix of the two that are admixed individuals. And here I'm highlighting A, B, C, and D as just 4 example individuals shown below in like kind of a standard structure plot that you might see from an admixture analysis. So from these analyses, we get admixture coefficients, also known as Q values. They range from 0 to 1, representing the relative ancestry from each population. And we can use this to calculate an admixture score, which is just 1 minus the maximum ancestry coefficient. So you can see for A and D, because they don't have any mixed ancestry, the admixture score is 0, whereas for B and C, because they have mixed ancestry, it's a higher value. And then we took all of these variables and we used a model selection procedure where we basically tested all the different combinations and then used AIC to select the best model. So what did we find? Well, the best model had 3 variables. Paleoclimate change, glacial extents, and admixture were the best explainers of genetic diversity in Salavoris. And just these 3 variables alone explain almost half of the variation in heterozygosity in this species, which is crazy. We're talking like whole genome across 14 million SNPs. We're able to explain this much with just 3 variables. So what are these variables? So first of all, paleoclimate change is the strongest predictor of genetic diversity in this species. So just to break down these graphs, on the x-axis you have paleoclimate temperature change, y-axis is heterozygosity or genetic diversity, and then this graph on the right is showing each of our samples colored by paleoclimate temperature change, just like the plots on the graph. And we see that with increasing temperature change, we have decreasing heterozygosity. What you also might notice is that this is a northward, kind of northeastward gradient. And if you remember, Bouzier et al. Found that there was this range expansion northward, which we think might be tied to this change in genetic diversity. So as you expand northward, you're having decreasing genetic diversity at the range edges. Also, this could be the result of population size fluctuations during the Quaternary glacial cycles because areas that experience the most temperature change may have had more extirpations or more recent colonizations. In a similar vein, areas that were glaciated during the last glacial maximum are cold spots of genetic diversity. So here, that blue part is just the glacier that's sitting kind of on top of the Sierras, and all the points inside of it are colored blue, and you can see they have much lower genetic diversity than the areas outside it. And this really lines up with previous work by Adam Lachey, saying that these higher elevation Scloporus species probably really recently colonized these areas post deglaciation. And then finally, maybe not surprisingly, but admixture creates hotspots of genetic diversity. And admixture, maybe like that's not a landscape variable, but it kind of represents where phylogeographic breaks are. So for example, we see that the spot where we have the most admixture and also really, really high genetic diversity is in the transverse ranges down in Southern California, which is a known phylogenetic break of importance in a bunch of species across California. But what about our contemporary landscapes? Do they really not matter at all? Well, this is where things get really complicated because we do find some contemporary variables seem to have strong relationship with genetic diversity. For example, contemporary temperature has a really strong positive effect on heterozygosity, which you might expect. They're lizards, they like it warm, these areas might host larger populations. However, the problem is that landscapes are correlated with each other. So these contemporary variables are significantly correlated with the paleoclimate variables, and that's why they end up getting dropped from our model. So this doesn't mean that contemporary variables aren't important. It could just be they have a weaker effect because these are more recent changes that we're talking about. But also it is possible they don't have an effect and it's all historical. So that's what makes it really challenging when you're doing these spatial landscape studies. How do you disentangle the two? And lastly, we were kind of curious, what does this mean for the future, right? So because genetic diversity is non-randomly distributed across space, it's structured by climate, what does this mean for how future climate change will affect current cold spots and hot spots of genetic diversity? So we find that future climate stability is actually much lower in areas of low genetic diversity. So here on the x-axis, you now have climate instability, so increasing values, more unstable. And then each graph going this way is a scenario of increasing climate severity. And you can see that the relationship between the two gets stronger and more negative the more left you go, or more right, sorry. But yeah, so we see that these areas where genetic diversity is low are also the areas that are going to be more unstable in the future. So where does this leave us? Well, one, we see that historical landscapes are really important for explaining genetic diversity, specifically paleoclimate change, glacial extents during the last glacial maximum, and admixture are able to explain almost half of the variation in genetic diversity in oxen talus. We also see that confounding correlations between historical and contemporary landscapes make it really tricky to disentangle contemporary drivers of genetic diversity. I think this is a really key point because when we're thinking about modeling things like genetic diversity, we really need to look at across time scales. You know, if you include if I included only contemporary variables, I might have been like, oh yeah, temperature, that makes sense, that is the main driver, but really paleoclimate change is what's probably driving a lot of this pattern. So yeah, considering multiple temporal scales is really key. And then finally, we see that future climate change can disproportionately affect areas of low genetic diversity due to the role of past landscapes in shaping current genetic diversity. And this has also been found in other species, like actually caribou in Canada, and something to think about because it's likely that a lot of other species are going to have genetic diversity that's structured by climate. So what are we doing next with this? Well, I'm looking now at genetic diversity through time, so using methods like SMC and PSMC to see if changes in population size over time collide with changes in climate. And then we're also looking at how landscapes shape adaptive variation, using a bunch of museum specimens to get phenotype data from CT scanning to look at local adaptation in this species. And that's it. I'd like to acknowledge all my lab mates and funders, and thank you all so much for coming. I'm happy to take any questions, and please come talk to me about any of this anytime. Anyway. Yeah.
Did you do any, by the way, great job, did you do any of that landscape modeling in conjunction with like historical and contemporary migration modeling, like V8 or anything?
Through what?
Like V8 or another historical migration model, or was it primarily focused around landscape features?
Yeah, it was primarily landscape features. The migration thing's interesting. I definitely I don't know if you've heard of the themes or Eames approach. That's something that I want to do next, maybe to see how migration plays into this. Bouzy et al. Did actually a lot of the great migration work on this species, which I didn't show here because I didn't have time, but showing that basically it seems to be, this northward expansion over the last 100,000 years.
This might be a little naive. So you spoke about paleoclimates where you're looking at a rate, right? Rate of change of temperature, one of the minor factors. And for current contemporary time scale, would you think taking in variability and not a rate itself would be something that could be taken in consideration for contemporary ones, because that's something that's more relevant than just a simple rate, like the variability in how environments are fluctuating, or maybe the ranges that can be like the range shifts or the differences in, let's say, temperatures that can be seen between years could be something that can be correlated. Can we maybe separate from your paleoclimatic rate?
So just to reiterate that question, you're asking whether variability, like between years variation, would be for the contemporary variables. That's an interesting question. I think I have to finish, but we looked at that for the past variables because we have climate fluctuations and climate stability. That's certainly important, but I haven't looked at that for contemporary that lasts 100 years. We do have climate change, which should approximate that, but that'd be interesting to include. I'll definitely think about that. Yeah. So we do have. The relative rates of change are based on contemporary last 100 years of change, but definitely variation would be interesting too. Sorry, I have to pass on the next person, but come talk to me later.
Hi, I'm Maya Wilson-Brown. I'm a PhD student in the Joseph's Lab at Michigan State University. And today I'm going to talk to you a little bit about local introgression patterns in Capsella bursipastoris from New York City. In order to do that, I do have to talk about polyploidy. I'm so sorry. But I think a lot of contemporary biologists appreciate polyploidy as a major driver of evolution. And one large aspect of that research is trying to reconcile the prevalence of polyploids in nature with well-known and characterized difficulties with newly formed polyploids establishing viable populations. And for a long time, people thought of polyploidy as a near instantaneous way to get reproductive isolation between populations, but we now know that that is actually not the case. There's a lot of empirical evidence of gene flow between populations of different ploidies. And so one such system in which we can study gene flow between ploidies is the genus Capsella. Capsella has 4 widely recognized species in the genus. Capsella orientalis, Capsella rubella, and Capsella grandiflora are all diploids, and Capsella bursipastoris is the only tetraploid in the genus. Capsella bursipastoris is also the only species in the genus that has a global distribution. The diploids have very limited ranges in Eurasia. There's also been a lot of previous evidence of gene flow from Capsella rubella into Capsella bursipastoris. So in 2008, Tanya Slata and colleagues found a non-zero migration rate from Capsella rubella into Capsella bursipastoris in Western Eurasia. And then again, in 2015, researchers found evidence of admixture between Capsella rubella, shown in green, and Capsella bursipastoris, shown in yellow and blue. And then again, in 2019, other researchers found even more evidence of admixture between Capsella rubella and Capsella bursipastoris in Europe and the Mediterranean. And so if we look at that toy phylogeny I showed earlier, we see something like this, where there's evidence of admixture from Capsella rubella into Capsella bursipastoris, but only in the populations that occur in Western Eurasia. But I also mentioned previously that Capsella bursipastoris is found everywhere, and I live in the introduced range of Capsella bursipastoris, so that's what we're going to be talking about today. So we might ask, how does variation in the introduced range of Capsella bursipastoris compare to variation in the ancestral range? And so in 2017, a former postdoc in the Joseph's Lab collected 24 populations of Capsella bursipastoris around the New York City metropolitan area. And we grew up those populations in growth chambers at Michigan State University and whole genome sequenced 2 to 3 individuals from 23 of those populations. And then we combined that New York City data set with publicly available whole genome sequencing of Capsella species and aligned all of these individuals to a newly generated Capsella bursipastoris reference genome in our lab. So here I'm showing you the results of an admixture analysis of Capsella bursipastoris in the ancestral range, and we get about 3 population clusters, one from northern Eurasia, one around the Mediterranean, and another one from eastern Eurasia. And these results are recapitulated in a bunch of other studies of Capsella bursipastoris' ancestral range, where we get about 3 population clusters with this data set. And so if we apply those admixture groups to the New York City population, we find that the New York City population is by and large derived from northern Eurasia. But there is a little bit of ancestry that might be inferred from both the Mediterranean as well as the East Asian population, and so we just wanted to dig into that a little bit more. Because we have whole genome sequences, one benefit is that we can do local ancestry inference, and local ancestry inference is just a way to determine ancestry at different loci across the genome. For this, I'm using Ancestry HMM, and it works this way, where you define your parental populations. Here in this situation, I'm trying to distinguish Capsella rubella ancestry from Capsella bursipastoris ancestry, and so my parental populations will be Capsella rubella and Capsella bursipastoris from East Asia, where there does not seem to have been any admixture from Capsella rubella. And then my admixed population will be Capsella bursipastoris from northern Eurasia, the Mediterranean, as well as New York City. And then you define allele frequencies at highly differentiated SNPs in the parental populations, and then compare those allele frequencies to that in your potentially admixed population. And then it outputs a probability of parental ancestry at each SNP. Just to orient you to the next few plots I'm going to show you, I have individuals shown as bars across the horizontal axis, and I will represent homozygous bursipastoris ancestry in green and homozygous Capsella rubella ancestry in red, and position along chromosomes on the y-axis. And then heterozygous loci will be shown in dark gray. Because I'm using a population of Capsella bursipastoris in order to define bursipastoris ancestry, I want to make sure that it is actually bursipastoris that I'm using. So I actually withheld 5 individuals randomly chosen from the East Asian population and added them to my admix group just to make sure that I wasn't getting any Capsella rubella ancestry inferred. And what we can see here is across all 5 individuals, across all chromosomes, and across all positions on those chromosomes, I'm only recovering Capsella bursipastoris ancestry, so I feel pretty good about using that population to define Capsella bursipastoris ancestry. And then if we look at the Northern Eurasian population, we do find Capsella rubella ancestry inferred in all individuals across all chromosomes. And the same goes for the Mediterranean population. We do have Capsella rubella ancestry inferred in all individuals across all of the chromosomes, which is great because other studies found that with reduced representation sequencing and also aligning to a different reference genome. And then when we look at New York City, we find much of the same, where every individual in New York City has some Capsella rubella ancestry inferred across all of the chromosomes. And if you squint a little bit, you can kind of look at patterns, but that is like a bad way to do that, so don't do that. And so I also wanted to be able to compare the populations to one another. And so if you think about the plots I've just shown you, and then think about them rotated 90 degrees, and then we're only focusing on the Capsella rubella portions of the genome, and then I squish all the individuals like this on top of each other. And so the color value now is indicating how many individuals in that region have Capsella rubella ancestry inferred. And so for New York City, we see quite a bit of variation both in the number of individuals that have a region as inferred as Capsella rubella ancestry and the different sizes of those regions. And when we compare New York City to the other potentially introgressed populations, we see very similar patterns across them. So regions where many individuals are inferred to have Capsella rubella ancestry in one population also have many individuals inferred to have Capsella rubella ancestry in another population. So just to give myself a little bit more confidence that what I'm picking up is actually Capsella rubella ancestry, I did principal component analysis at different locations and windows along the genome. And so in this example, the window I'm showing is in the gray bar, and no individuals in any population have Capsella rubella ancestry inferred in that region. And when we look at a PCA, that does seem to be true. So all Capsella bursipastoris individuals that are different colored circles cluster together, and they're importantly not clustering with the pink triangles, which are Capsella rubella individuals. And as another example, in this window, none of the New York City individuals have Capsella rubella ancestry inferred, but a few of the Northern Eurasian as well as Mediterranean individuals do. And when we look at the PCA for that region, we see exactly that. A few Northern Eurasian individuals and a few Mediterranean individuals are clustering with Capsella rubella in this PCA. Even more importantly, they are not clustering with the other Capsella species, so I feel pretty confident that what I'm picking up with this method is actually Capsella rubella ancestry, as opposed to ancestry from another Capsella rubella species, or Capsella species, sorry. Okay, and then for some last little tidbits, we did find 28 introgressed regions that were only found in the New York City metropolitan population. On average, these regions are pretty small and only shared by a few individuals. So for some conclusions, New York City Capsella bursipastoris derives a majority of its ancestry from Northern Eurasia, and the patterns of introgression vary within New York City. And we were also able to capture additional variation in New York City that was not captured in the ancestral range. With that, I would like to thank my lab, especially Adrienne Platz, who helped generate the reference genome that we're using here, Rebecca Penko, who did all of the collections in New York City, my funding sources. You can email me, or you can email Emily, or you can just Google me. And with that, I will take questions. So the question is how I managed to work with both a tetraploid and a diploid. In these analyses, I'm actually only looking at one subgenome of Capsella bursopastors. So Capsella bursopastors reads were aligned to both subgenomes, and then all of the diploids were only aligned to one subgenome of Capsella bursopastors. So I'm sort of treating it like a diploid because I'm ignoring the other subgenome. Yeah. Thanks.
Really, really cool. Really nicely done. This is a quick speculative question. You know, if you have these progressive regions, what do you think is and you know, there's some pretty small regions in the genome.
Yeah.
Do you think, what do you think those regions might be? Do you think they're historical just contingencies, or that they're adaptive, or
Yeah.
like, yeah?
Yeah. So the question is what are these regions? I don't know yet, and that is like going to be the next steps of this research, is both figuring out what is underlying these regions and if they have any sort of like effect on the fitness of Capsella bursopastors. And they also have very different sizes, so that might indicate that they are of different ages, or it might not. So yeah, those will be the next steps. Okay. Thank you.
Okay, let's go. Hello, everyone. Thanks for coming. I'm Théo Gaboriau, a postdoc at the University of Lausanne. So I put that title, Geography of Constraint on Climatic Niche Evolution, because when I submitted the abstract, I was not sure exactly what I was going to turn on. But I'm going to talk about tropical niche conservatism. That's a long-standing hypothesis that is based on a main observation that species that evolved in the tropical environment during geological time, they underwent way less variation in temperature at geological time. So the green line is the variation at temperature at the equator, and the blue line is one at 40 degrees of latitude. So you can see that the amplitude of temperature along the geological time that the temperate lineages have faced is much larger. And if you look at the annual variation on the same latitude, it's the same pattern. The tropical species they undergo far less variation. That is about temperature. But the tropical niche conservatism concerns also hypothesis concerns also other climatic variables. And so because those species have been in a much more stable environments over geological time and also along the year, potentially they can specialize and find some crazy adaptation that are highly costly energetically, like flying super hard to stay at a stationary level in hummingbirds, or just deciding to live in a venomous animal like the clownfish. And those really specialized adaptation, they kind of, because they are like really costly, they kind of like prevent you then to like shift towards a new environment. And so because of that, you are specialized on something that specializes you in terms of climate niche as well. And so it has like several, this tropical niche conservatism hypothesis had several predictions. And those are the predictions that I'm going to talk about today. First, it predicts that there is asymmetric transition rates in favor of tropical latitudes. So the prediction is that there are more like species colonizing going towards the equator than going toward the pole. And the other one is more like really evolutionary hypothesis, and that's the temperate and tropical lineages inherit differential evolutionary constraints. So one is the rate of evolution of the climatic niche. The prediction is that the tropical species will have a slower rate of evolution of the climatic niche, and the other one is a constraint on the climatic niche breadth, and that's there is an evolutionary constraint towards smaller climatic niche breadths in tropical species than temperate species. So for the first prediction, what I did was use the major vertebrate superphylogenes and then try to also characterize which species are like tropical species, which species are temperate species, and also which species are widespread species, so both occurring at the tropics and outside the tropics, using occurrence data, and then using phylogenetic comparative methods, so here the SSE method with hidden states, to just calculate the transition rates from the tropics and from out of the tropics. So you can see here the first row is the transition rates to the tropics, and the second row is the transition rates from the tropics. And you can see that like my estimation like give for all the major vertebrate clades. So I did it on mammals, amphibians, squamates, birds, elasmobranch, and fish. And you can see that is consistent across the vertebrate clades that I studied, that the transition rates towards the tropics are higher than transition rates out of the tropics. So that does not concern yet the evolution of the climatic niche, right? It's only like transition rates. So to go further on that and on those questions and like test the other predictions, I then characterize the climatic niche all of those vertebrates. So for fish and elasmobranch, I just like focused on the marine species and used bio-oracle environmental data, so 26 bioclimatic data, bioclimatic variables to characterize the climatic niche of those species. And for every species that I could study, I got as much occurrences that I could from GBIF and INAT, and of course as much like correct occurrences. So it took a lot of filtering. I'm not going too much into the details, but a lot of filtering to sort out that data. And same for land, so for amphibians, squamates, mammals, and birds. I used the WorldClim V2 environmental data and gather the occurrences, and that led me to the characterization of the niche space for all of those major vertebrate groups. So here I show the sample size or the number of species for which I've characterized the niche. In green the tropical species, in blue the temperate species, and in yellow the widespread species. And well, that's not really useful to look at that. Expect that like from a rough visual point of view, you don't see so far that the temperate species take up much of the environmental space, except maybe for fish, but otherwise not so much informative. So we can try to characterize it a bit more, like in a bit more detailed way. So the first variable that I want you to look at is the niche hypervolume. So because I've like characterized the niche with the PCA using a lot of variables, then I calculated the volume of the niche of each species using a hypervolume approach, and looking for mammals, amphibians, squamates, birds, sharks, and fish. Actually, what we observe is that is not as expected. The hypervolume of the temperate species does not seem to be higher than tropical species, but more expected than the hypervolume of the widespread species tend to be larger, which makes sense. When you wanted to look at the latitudinal trend in hypervolume, so I took all of the species occurring at chunks of latitudes and calculated the mean hypervolume of species occurring at that latitude. We have a really like weird pattern, I would say. That's like kind of hard to explain what's going on here. But you don't really like see strong evidence that the hypervolume of species that occurred in the tropics are like much slower, even though we have like a decrease here, but it's not as strong. So not as expected, the hypervolume is not like smaller in temperate lineages, but well, it's really surprising, like considering like temperature, the temperature data that we have, where like there is really like much more temperature variation in the temperate climates. So when we look at temperature variables, we can recover that difference. So this is the annual temperature amplitude, and the temperate species like face much more annual amplitude. And also, this is the species temperature range, so it's like the range of temperature in which the same species can be found. And this also like much higher for temperate species than tropical species. So what about now trying to make some evolutionary sense into this? Is what we observe here is consistent, like evolutionary speaking. And to that end, I used a model that we developed in the group, the Jive model, that is measuring both the evolution of intra and interspecific variance on a phylogenetic trees. And so basically, this model takes individual level data and is trying to sample the modes of the phenotypic distribution. So now in our case, it will be the climatic niche. So just points on the PC axis of the climatic niche, and then apply independent trait evolution models on both modes of evolution. So here, for the climatic niche, what I did is trying to use this model to model the evolution of the position of the niche of each species, and at the same time the niche breadth. Why is it important to use that kind of approach? Is because I'm sampling from individual data, then I can have I have the variance of the climatic niche rather than just the mean or calculating the breadth. So with the amount of data that I have, I can correct for the sample size, and also using a variance instead of a range to estimate the niche breadth seems more relevant because you can really implement the preference of the species and the optimal value with the variance approach. And using this model, you can test different models that have differential rates of evolutions, and also models that have different optima, and so that allows me to test the prediction of the tropical niche conservatism. And so, what are the results in that end? We observe that usually the best model selected for the niche position is a model with a single rate of evolution and no really a constraint or optimal niche position. For all of the clades, we just have a differential rates of evolution for the squamates, but otherwise the niche position seems to be really unconstrained by having evolved in the temperate climate or tropical climate. But then when you look at the niche breadth, the variance, you see that for a lot of clades and a lot of variables, you have differential optimum for between the tropical lineages and the temperate lineages, which shows that there is evolutionary constraints on the climatic niche breadth, but not really differential rates of evolution of the climatic niche. So that difference is that what we expected? Not really. So if you look, I cannot show all of the PCs and all of the major clades at the same time. So I decided to show one marine clade, one terrestrial clade, and one clade that is also exotherm. So mammals that are endotherms, we show actually the opposite prediction of what I thought. Meaning that when you look at the axis, you see that the optimal niche breadth of mammals is larger in the tropics than in the temperate, which kind of makes sense with the hypervolume data that I showed before. Same for the elasmobranches. you can see that the optimal niche breadth for the tropical species in sharks is much higher than the optimal niche breadth for the temperate species in sharks. But when you look at amphibians that are ectotherm, that's the opposite patterns. We can see that the temperate species have a much larger, well, have a larger optimal niche breadth. So, like to conclude about that, we found like mixed results concerning the predictions of the tropical niche conservatism using like a really big data set on vertebrates. We do find asymmetric transition rates in favor of tropical latitudes, but we don't find a lot of differences in rates of evolution of the climatic niche. And we also like find climatic niche, evolutionary constraint on the climatic niche breadth that is not exactly the one that we expected before. So with that, I would like to thank my lab and also thank you for your attention, and I'm open to questions.